Our next laureate, our next speaker, is Dr. John Jumper. So John Jumper was born in 1985 in Little Rock, Arkansas, USA. And he received his PhD in 2017 from the University of Chicago, Illinois, also in the United States. And he is at present senior research scientist at Google DeepMind, London, UK. So please join me now in welcoming Dr. John Jumper for his Nobel lecture.
So it's absolutely wonderful to be here. It's truly an extraordinary honor to tell you about this work, to tell you about the work of our team in protein structure prediction. And the part that I'm going to focus on today in my lecture is how did we think about building chemistry and physics and biology into this neural network? How did we take AlphaFold from kind of standard machine learning into trying to build the machine learning of proteins, and why did it matter? And kind of a starting point for this, and really the starting point for my entry into biology, is I applied for one job and ended up getting another, and ended up working in this lab that worked on protein simulation, D. Shaw Research, and really thinking about how proteins move from a very, very direct kind of brute forcing Newton's equations type of perspective. And there's such a direct way to put the physics into it. And so you think about these atomic systems, this kind of collection of atoms, you kind of throw away the electrons and try and replace them with still a good energy model that can be very, very directly used in order to compute forces, in order to update the positions, and to watch proteins move. And it's absolutely mesmerizing. And it's normally what people kind of think when they say, let us put physics into our protein computation, right? Let us think about the forces on atoms. Let us reduce it to, you know, as a young physicist, right? Your hydrogen atoms, your really well understood kind of physical principles, your van der Waals interactions, your Coulombic interactions. And people write down these kind of equations like you see on the right. This is from Michael Levitt's talk, and really a wonderful depiction of how people think about putting physics into simulation. And we have these kind of nice laws of motion, and if you look at it, you can see things, if you're a physicist, that you really recognize. You say, there is the physics. But if you look really closely, you might realize how much physics is missing. That all of these interactions are pairwise. For example, this is a non-polarizable model. What I love most about the depiction of the equations is that it is almost written in Comic Sans, right? It's written as if it's a kind of coarse and approximate description of the physics, which of course it is, and it's told us a lot. But there is so much more physics. It's really more like a machine learning model of a very particular and effective form, but that is very, very expensive to use. And it is a really interesting way to think about some of the physics, but maybe not others. And it's very, very deeply difficult to reach the types of timescales that we need in order to make predictions of processes like protein folding. And more importantly, even if you can drive one simulation out to the millisecond plus scale on which most folding occurs, you'll find that your model wasn't quite right, right? This is still a cartoon of the physics in some sense, fit with many parameters. And you need to change those parameters in order to make it more accurate. You need to fit this machine learning model, but it's so very expensive that it's very hard to do so. And so, in fact, if you look at a lot of what has been successful in protein structure prediction prior to AlphaFold and through AlphaFold 1 has been a machine learning point of view, and really in a homology point of view as well, that starts from this amazing resource, starts from the other side from the physics. We start from the observations, and of course it's the protein data bank. And both David and Demis talked about how essential it is, so I'll assume you're convinced by now that this is a tremendous resource, and a tremendous amount of foresight went into it, making it available, collecting the full output of the structural biology community, and so that we can develop computational and test computational methods, and it grows at a rapid pace, and grows at enormous investment. You know, I look at the left-hand side, we've crossed 200,000 structures in the PDB, and each one is something like a year of a student's time, someone that we're going to call doctor at the end of it. It's a truly incredible societal investment in these data, and it gives us another way to go after this. We can, of course, use machine learning, and there have been many approaches to develop machine learning for protein structure prediction. And one of the most successful paradigms would start from the other big source of information we have, protein sequences, would convert that into coevolution statistics, what type of residues varied together, and then use those in order to try and make some predictions about the structure that would then be fed into an engine to kind of turn it into a proper structure. And our AlphaFold 1 system was a very good example along this. And it uses a neural network that was really almost taken off the shelf. I think quite literally the same neural network design as was used for a computer vision project within DeepMind. And what it has is biological inputs, coevolutionary data, sequence data. It has geometric or physical outputs, the distances between proteins. And then, of course, we make many distance predictions in squared. So for 100 residue protein, we're making, or n squared over 2, we're making 50,000, if I did that math on the fly correctly, 5,000 predictions of protein distances, and then we try and harmonize them through a kind of really, we ended up with a very simple procedure. We had very complex procedures. There's a lot of kind of Leventhal-inspired search, and how do we really do search? Well, it turns out it was kind of gradient descent in the end. And I think what was interesting as we looked into AlphaFold 1, we had built it. It was a great system. It had some new ideas in terms of distances, but there was something actually really interesting and curious about it as we looked deeper. That sometimes AlphaFold 1 was very confident of the distances between different parts of the protein. I think the view going into this, or how coevolutionary data is thought of maybe giving us some information about contacts. It was originally showed that close-by residues tend to evolve in a coordinated fashion, and then it was shown that, oh, well, using these data, you can sometimes find out close residues. But AlphaFold 1 would sometimes be incredibly confident that 2 residues were 13 angstroms apart, and that's quite a distance. That's kind of like saying, I am really sure that my friend is 178 meters away, right? You don't know that just by saying, I heard them. You know that because you know exactly where they are, and then you say it's 178 meters. And so similarly, we had some indication, even in AlphaFold 1, that maybe sometimes it was solving local parts of the structure. Understanding the geometry really well, and then it was reporting distances by basically measuring the geometry of this thing that it understood in the middle and had no way to tell us. So we had this kind of sign that there was something going on, that maybe these neural networks could do quite a bit more, but we were also held back by, you know, bringing together 2 fields with an awkward interface, right? We had the incredible field of machine learning and all the ideas there, and then kind of in and out, we had, you know, biology, geometry, but we didn't ever meld them. They weren't working together. And because of that, all of the understanding of geometry, biology, et cetera, well, almost all of it had to be learned from data, had to be learned from these, I think at the time, about 140,000 protein structures that we had available. So we were entirely dependent on the general learning capabilities of our general learning systems, and that's great, but that's not very data efficient. How can we take all of this kind of human knowledge that we have and build it into the system? And kind of the first stage of that is we wanted to get to something incredibly simple, not a, you know, you do all this processing and then you do all that processing and you have a little bit of machine learning in the middle. How do we just make it, you know, this simple diagram? We'll start with a sequence on the left, we'll do a big neural network thing in the middle, and then we will end up with a structure on the right. And kind of getting to that really felt like it was going to be essential. We needed some way to do this, and we needed that thing in the middle to be inspired by physics and chemistry and geometry and really build those ideas into the design of the network so that it could be more efficient with the data we had. And the way that we started doing this, and this was really shortly after AlphaFold 1 in late 2018, we started to say, well, let's just throw away the evolutionary information. It's good, but we really want to understand physics. We really want to understand geometry. And so we kind of went back to the beginning. And also, it made our performance on almost all proteins terrible. And starting with terrible performance gives you a license to try weird ideas, because then your weird idea can be state of the art. And the other thing it did is it started to really get us thinking about, well, what is in proteins? What are in these protein chains? And I remember I'd been working with, even back into my PhD thesis, this notion of geometric frames, that each residue has a side chain sticking out of it. And that residue is an important, that's an important thing, where this points. How do we put orientation into it? And we started doing what we would call frames or reference frames. And we started to play originally with side chain prediction. And oh, can you predict where side chains are? And actually, you can do that really, really well with deep learning. And then we kind of thought more and more, well, how much further can we go? And we tried some ideas that looked more like kind of learned simulation. But then we found out, actually, that if you take an architecture that has some understanding of geometry, if you put some of this into the neural network, you can start to produce protein structures directly. You can put in a sequence, and you can say the answer is this structure. And there were a couple of essential innovations along the way. Probably the most important was simply how you measure how correct your answer is. And in fact, we came up with the FAPE loss that says, how are these geometrically and orientationally related? And this started to work reasonably well. And actually, I remember before the first kind of direct structure prediction that worked, I had some idea that that actual folding, making structures, was going to be an imagination for the network. And so if you see the diagram on the bottom, this was essentially the diagram we had at the time, is maybe we'll do embedding, we'll think about the sequence, then we'll do a bunch of folding, and then we'll think about it some more and think about what we got, and we'll do it again and again. It ended up actually looking a lot like recycling, and I didn't appreciate that until we put it into this talk. But we had this idea of we're going to build geometry as a kind of tool that the network is using in producing structures. But what we found is, in fact, that little folding bit already worked really surprisingly well. The answers were still really bad, but they looked like proteins. It was clearly handling the geometry of proteins immediately. And then only a week or two, maybe a month after this started to work, Rich had the idea of, well, we have all of these distance predictions from AlphaFold 1 sitting around. Why don't we just stick those into this geometric structure prediction network as well? And it looked a lot like the structure module, in fact, for the final AlphaFold 2. And so we would stick distograms into this and almost immediately got predictions that were nearly as good as AlphaFold 1. And so we had nothing but neural network components now. We had kind of stripped at least the geometry off the end and gone to what we knew we needed, the protein structure. But we still had the coevolution, in fact, the fixed network at the beginning. And of course, we tried many, many ideas for new geometric networks, and all of them were about the same accuracy. We pushed it up only a couple of GDT over this research and started to refer to what we called the distogram barrier, that we weren't going to get any better at this problem until we got better at making the distograms, until we eliminated our dependence on the AlphaFold 1 network. And in the same way as we had kind of reimagined how we built and built new components for the network for the end, we wanted to build new components at the beginning. We didn't want it to be dependent on both coevolution and this interpretation of coevolution in the pseudo-likelihood models. To do that, we started over just, and this was actually, we first tried what has now become popular, but we didn't really do big enough or deep enough versions of it, single sequence prediction. Can we just fill in the protein sequence and train models to do that and have that be some sort of way in which the model learns about sequence space and then go and predict distances? But pretty immediately we said, well, the multi-sequence alignment is still great. This still is telling us a lot about the evolutionary history of the protein. We know that it's highly relevant to structure. How can we use this? And so we started to train networks only on this, and in fact do what would be considered mass language modeling training now. We would call it BERT at the time. And we would just put in the raw MSA with a number of blanked out sections and said, guess what amino acid is here? And to do that, you have to understand a lot about both the evolution and the structure in order to guess which amino acids might be in the hidden locations. And this was a raw MSA kind of BERT network, BERT being the first major mass language modeling, so we called it Robert. And we also, I think, made a really consequential choice here, that the multi-sequence alignment, the collection of many, many protein sequences aligned such that each column is an evolutionarily related location, we decided to use axial attention, attention being a way in which the whole sequence can talk to each other in some sort of averaging way. And this was semantic because we could say the sequences meant something. So attending over them is attending over different tips of the evolutionary tree. And the columns meant something different. They meant kind of attending over this position over evolutionary time or across evolutionary endpoints. And alternating those two is a very, very semantic way to think about processing evolution. And this worked extremely well. And so as soon as we had these and they started to work and they looked interesting, we kind of took the same convolutional networks and just basically attached it on top. And so we would run our MSA, it would try and fill in the MSA, and then we would go send on what had been learned to this convolutional network we developed and predict histograms. And this worked exceptionally well. Suddenly this network was learning incredibly quickly. We had broken the histogram barrier. And almost immediately after that, and so of course we would put this into direct folding, but almost immediately after that we said, well, okay, we've gotten some new interpretation of how to process evolutionary data. We've aligned the neural network to do that. Why don't we just attach that to our view of geometry? And so we would put the structure module on it, and we suddenly had end-to-end learning. We would start at almost the raw inputs, the aligned evolutionary data, and then we would produce a structure. Now, we asked it to do two subtasks along the way. Oh, by the way, fill in the MSA. Oh, by the way, make a histogram to keep the network, to give it, lots to think about, lots to do. But we almost immediately had end-to-end structure prediction in kind of early to mid 2019. And it became really clear that this was a great idea, that this also, it meant two things. One is that it was more accurate, it was more aligned. It also meant that it was very, very easy to train and test new ideas. And so we could have this incredibly creative period where we could try many, many ideas and, find out every day that your idea from yesterday was wrong, right? And try each of these. And machine learning is filled with failure. You have intuition and failure, much like lab experimentation. So you need to try many, many ideas to make it work. Now, the other thing we actually realized pretty quickly, and I was reading a paper on sparse transformers on how you can use attention on images, and they said, oh, well, it's a good idea to do axial attention, across the rows, down the columns. And we're sitting and reading that and realized, in fact, that the second part of processing, where we dealt with in-residue by in-residue matrices that represented kind of all pairs in the network that would end up being the histogram, well, in fact, that is a very semantic operation. And if you think of it as a graph, we can attend all of the residues that relate to this one particular, or all of the pairs which attend to this one particular residue in either direction. And this was very semantic. And even better, we had already coded that up because it was good for the MSA. So we started to replace the convolutional networks with axial attention. It was much more aligned also because if you think about pairwise information, well, physics is pairwise only in its energies and only approximately, but the question of structure is highly non-pairwise, right? Things come together. There are many, many pair interactions, and you need all of those talking to each other, communicating. And so axial attention lets them communicate incredibly quickly, lets the network constantly refine its hypothesis rapidly. And the animations that Dimas showed of the structure prediction over layers, you can see how quickly it's changing its mind, it's updating its view, and axial attention is essentially what allows that, and that started to really drive enormous accuracy improvements. Now, and I'll only describe a couple of the ideas we used in the middle, but as soon as we had done that, we started to say, well, gee whiz, this process of putting physical and biological intuition into our neural networks is a great idea. Let's do more of it. Let's try all the ideas we can, always guided by what made our structure prediction at the end more accurate. How can we find new ways in order to orchestrate information flows that reflect our understanding of physics, that give the network the opportunity to do something more aligned with our understanding of physics? And so two of the really great ideas one from Michael who said, well, you know, this whole thing of processing the evolution first and then trying to do the structure second, that doesn't entirely make sense, right? You want to think about the evolution in light of the structure, and of course you want to think of the structure in light of the evolution, so why don't we just process them both at the same time? And we have this clever scheme where the evolutionary information, the MSA over here, would come in, and as we were processing the sequences in the MSA, we would bias that information by what we believed about the structure, by making a connection into the structure. Similarly, whenever we learned something new about the MSA, we would drop that back into our representation of pairs or structures, and we created this conversation between what we would call two towers, but I've written it sideways, of the network that allowed it to engage in more precise semantic processing of this. And another idea that was come up with, this was one from Alex, is how do we, you know, well, when we think about processing pairs of residues, right, you have, say, I and J as two residues, and then you might have I and K as another two, and those two are talking to each other. But what about the third edge of the triangle? What if we just put that in? And so we started to put in what we would call triangular attention, and very shortly after triangular multiplicative updates, and let us just build this also because we were inspired by this notion of, well, all of those have a strong relationship. They have the triangle inequality. There's something about all of those distances that must be interrelated. Let's create the connections that let them talk, let them compute about this, and in fact, you can measure afterwards that including these triangular connections makes the final distance predictions more consistent with geometry, about fourfold greater agreement with or reduced violations of the triangle inequality by putting in these connections. And so we're always building, not literally, we're not writing the triangle inequality and forcing the network to use it, because that doesn't work very well. We're building the connections that make it more efficient to learn the things that we know. Now, as we, in fact, in 3 stages of the project, we came to this problem of it's not just about structure prediction, it's also about knowing when you're wrong. And the first time we came up on this was a clever idea of Rich's of, oh, well, let's also train AlphaFold on AlphaFold predicted structures, but just the good ones. And as soon as you say, but just the good ones, you have to say, well, how do I know if a structure is good? And the original answer, and from some other work done by, I think, Tim and David, is, well, why don't we figure out how much AlphaFold's distogram is very particular and confident? And we called this a kind of KL loss. And so we would filter and only use those structures or parts of structures where AlphaFold was really sure of what it was giving. Now, as we got further in the project, in fact, shortly before CASP, we made predictions for SARS-CoV-2. And we said, oh, well, we really need a loss function that tells us, you know, or tells biologists what parts of these predictions can we trust. If we're going to make predictions from SARS-CoV-2 with our, you know, still secret AlphaFold 2 system or almost AlphaFold 2, we better tell them which parts are right. And so, and we said, well, all we really want to know, and thinking about it is, you know, we want to know the correctness, which is typically measured by LDDT, is one of the measures of correctness. And so can we just predict that? And we put almost nothing but a head that says predicts LDDT. And sure enough, that was extremely accurate and became incredibly important after we released. I remember biologists talking about this on Twitter, and they very quickly noticed these confidence measures are really, really reliable, tell you when you should use these structures, and started to say things like, if it's blue, it's true, where blue is our confident color. And it's so important because we can't really afford to have unidentified hallucination. You can have hallucination if you say, I don't think this is right, or maybe this is right. But you really do need to tell biologists who are about to do days, weeks, months of experiments based on what they just saw. You need to tell them if your network believes it. And the final confidence measure actually came up due to a reviewer comment who said, well, predicting LDDT is good and all, but that's a very local measure, and I think TM score is a lot better. Can you predict TM score? And started to sit down and write, no, that's not how this works. And it's like, well, you know, if we predict this and predict these kinds of measures and predict a pairwise measure of whether things are correct, then actually we can tell you what the TM score is predicted to be. And more importantly, this pairwise matrix tells you, a big complex neural network prediction isn't right or wrong. It has a set of hypotheses. Do you really believe these two parts of the protein are near each other? And so we could report that. And I think one of the kind of great and pleasant surprises of the AlphaFold release is how well biologists have used these notions of confidence to inform their research. How much they really do kind of not trust entirely, try and trust in proportion to how it should be used, these systems, and really deploy it responsibly in their research. And I've been so impressed, and they've been so good at that. It was something that I was very worried about. Would they simply trust everything? Right? These are predictions. Now, there are many, many other ideas I haven't told you about. I listed some up here. There are many, many more. Ask the AlphaFold team members in this audience to tell you some great stories. But which parts of it matter? And so after we finished and we were writing our paper, we said, well, can we just knock out ideas and see which ones matter? In the machine learning community, this is called ablations. And so as we moved ideas, I remember we had trouble breaking AlphaFold 2. We found every idea was worth something, and no idea was dominant. And I think this really reflects that there isn't one silver bullet of bringing physical and chemical intuition into the network, that there are many, many ways in which you align it, and every one makes you more efficient. In fact, Mohammed al-Qureshi's lab trained an AlphaFold 2 reproduction on about 1% of the PDB and found it as accurate as AlphaFold 1. That building these intuitions into the network is about worth 100-fold in data. So we are amplifying our usage of the precious experimental data we have by 100-fold by building these ideas in, by doing new types of machine learning. We, of course, make this available in partnership with EBI, who've been incredible partners in making a huge number of structure predictions available, enabling new research. I love research, for example, that finds an unknown density in a experimental cryo-EM structure and then searches through AlphaFold database to find, oh, well, this protein matches in shape and confirms it with biochemical experiments. There's so much more we can do, and we're ultimately closing what has been an enormous gap. That protein sequences are discovered at about 3,000 times faster than protein structures are determined. And we need machine learning to close that gap, and also to bring kind of the importance of protein structure not just to humans, but people that study plants, people that study pathogens, all of those different underinvested areas. And I think it's so exciting to see people use it. Now, finally, and I want to acknowledge very, very much, this is the work of a team and the best team that I've ever worked with. And I decided not to use the pandemic one of us all as squares on Zoom, but for most of the work, it was shoulder to shoulder. And these are really a wonderful group of people, and if you get a chance, ask them to tell you some of the stories. They are truly wonderful. And I want to acknowledge everyone who contributed. There was really a huge effort across many areas. The core team was about 15 or 18, but the support we got from the rest of DeepMind, from the wider community, was exceptional. Our collaborators, of course, at EBI, CASP, who not just kind of assessed and showed the world how you should assess computational science, but really helped to define the problem. Brought us GDT and other measures that really, really matter, and our research is deeply indebted to them, showing what works and showing how you should measure it. And of course, the experimental community, and I hope some of the people in this room on whose structures we trained. Thank you.
Okay, please join me now in welcoming all three chemistry laureates up on stage.